In this talk I'm going to be introducing you to experimental studies. It's the first of 3 talks on these studies. So going back to the hierarchy of study designs that I introduced you to not long ago, we can see that experimental studies or randomized trials are right at the top. So they're really regarded as the gold standard of epidemiology studies. And the topics I'm going to be talking about in my 3 talks are these. Starting generally, we'll describe the parallel study design, which is the most common, issues of randomization, blinding, how to improve compliance, a bit on data analysis, and some alternative study designs to the parallel study design, and finally ethical issues and when studies should be terminated early. So those are the topics that will be covered in these 3 talks. So the defining feature of experimental studies compared to all other types of epidemiology study is that the investigator allocates the exposure, or treatment as it's sometimes referred to in experimental studies, and the allocation is randomized in some way, and we're going to talk about methods of randomization. And so people are basically, from the recruitment, they're followed over a period of time and observed to see whether the study outcome of interest occurs. And they go under other names, sometimes called intervention studies, and then depending on whether it's a treatment study or a prevention study, as we'll talk about, there's a difference. There may be randomized controlled trials, randomized clinical trials, therapeutic trials, and prevention settings. They may be field trials or community intervention trials. So various terminology used to describe these studies. As I said, experimental studies are right at the top of that hierarchy. They're the gold standard of epidemiologic research, and if we can conduct them, and we can't always conduct experimental studies, but if we can, they're carried out well, then they're free of bias, and they can detect very small but very important effects. And sometimes you can think of them as being similar to say, toxicology studies carried out in laboratories with experimental animals, where groups of rats or mice, say, are randomized to different treatment groups. So it's the human analog of that. Now the primary requirement, the primary ethical requirement, is that you can only carry out such a study if the treatment or exposure, whichever you prefer to call it, has at least potential curative or preventive benefits, and more so, potentially at least, more so than the available alternatives. So this rules out many things such as cigarettes, alcohol, and so on. We can't carry out experimental studies with those exposures. So that is the primary consideration. Now the general conduct of these studies is fairly straightforward. We have to have a hypothesis, design a study, obtain funding, and of course get ethical or IRB approval. And then we go through a recruitment phase, and there's usually some specific eligibility criteria for recruitment. Talk a bit more about that, depending on the nature of the study, of course, and then informed consent is obtained from the participants. And then they're provided they're willing to be randomized, then they will be randomized to 2 or more treatment groups. And so the treatments may be 2 different treatments, or maybe 1 treatment and a placebo, for example. And then you follow the groups in time. Hopefully they take the treatment, and you monitor them, and depending on the outcome of interest, might be a cure, or might be the first occurrence of a disease, or recurrence of a disease, or could be side effects. And basically the statistical analysis involves comparing rates of the outcomes in the treatment groups. As I mentioned, the interventions tested can be preventive or therapeutic. So we can have prevention trials or therapeutic trials. And so an example here of a prevention trial is whether tamoxifen lowers the incidence of breast cancer in high-risk women compared to high-risk women not given tamoxifen. And such a study has been carried out, and I think it shows that women with a certain genotype do benefit from tamoxifen. Now, therapeutic studies, an example here, whether or not combinations of AIDS treatments or antiretroviral drugs are more effective than just a single drug for preventing or survival of AIDS, increasing survival of AIDS patients. Now, the study size, as we'll talk about in a future talk, we calculate the study size based on a number of parameters, and we carry out statistical power and sample size calculations. And there is software readily available, including Stata, can be used to carry out sample size calculations. Although there is other software which may be slightly easy to use in some ways for that purpose. And one of the things that determines the sample size is the number of expected events, the number of expected cures or number of expected new events occurring in the groups, and that figures very much in the calculation. And for that reason, because in prevention trials you expect a much smaller number of events relative to the say the study population than you would in a treatment study. For that reason, prevention studies usually have a much larger number of participants, so that they can detect the benefits or whether there's a difference between the two groups. So vaccine trials, for example, usually involve many thousands, maybe tens of thousands of people, just so that the effectiveness of the vaccine can be measured, because the numbers of cases of disease that will be prevented might be quite small relative to the population size. We can carry out these studies in both individual people and also in communities, but when we carry out community trials, they are inevitably prevention only, and an example of that would be fluoride in the water supply. Does adding fluoride to the water supply of some communities decrease the dental caries rate compared to communities where the water supply is not fluoridated? But studies with individual persons involved can either be therapeutic or preventive trials. So in a study, an experimental study, we can define several levels of population. So first of all, there's the reference population. Now this is the general group to which the results of the trial are intended to be applicable, and it may be as much as all people, or maybe a more restricted population. And then we sample that population to get an experimental population, which we will consider for enrollment. And they should match the intended reference population. And then we have actual study participants, those who actually participate in the study, and they were eligible and willing. So you can look at it in this way. So here's the reference population at the top, and then from that reference population we derive an experimental population who satisfy certain inclusion-exclusion criteria, as I'll talk about. And then some of those people will not be willing to participate, so we call them non-participants. So it leaves us with a population of participants, and we randomize at this point to the treatment group and the comparison group, which may be just two treatment groups, treatment A and treatment B, of course. So we do apply some selection criteria, and it may be that we want to pick people who are more likely to have the outcome. So if we're doing say a cancer prevention study, we would tend generally to pick older people, because they're more likely to have the cancer. So probably any prevention, such as tamoxifen perhaps, might not be so it would be more difficult to demonstrate its effectiveness in a younger population, just because the cancer events are so much rarer. And we also might exclude people likely to be lost to follow-up, and so people such as homeless people, alcoholics, and so on, or people who are known to be thinking of leaving the area might not be included. So that might be an exclusion criterion. And these selection criteria are applied at this stage from selecting from the reference population to obtain the experimental population. We can define several trial phases for new therapeutic drugs. The first one is a very small study, phase 1 study, where a few healthy people administered the drug at very low levels and maybe radio labeled, and in order to study how it's distributed in the body, how rapidly it's excreted, what the metabolism is, how it's changed in the body, and so on. And generally to focus on safety, not on its efficacy for any sort of treatment purpose at this stage, but just to see whether it's a reasonable thing to administer to people. And the second phase is a slightly larger study with different dose levels, when the focus is on both safety and efficacy. At that stage, the different dose levels will be evaluated. And then the phase 3 trial may be at a fairly large scale, maybe thousands of people, and these are the trials that are required for FDA approval in this country. And you may test it against the standard therapy currently being used. So most of what I talk about in these lectures are the phase 3 trials. And then finally there are phase 4. These are after a drug is being used. They have some sort of post-marketing surveillance to look for side effects and whether it's really effective in actual practice. And anyway, so these are sort of follow-up studies. Now, the most common design is the parallel design, and so each participant is allocated to one or the other of the groups, and they receive only one treatment. Now, there will be a comparison group with the treatment of interest, and so the comparison group will either get the current standard treatment or they'll get a placebo. Now, placebos are much less common these days just because there are current standard treatments for most conditions. So generally it'll be the new treatment versus the older, more accepted treatment. And the groups are followed up and in a very consistent way, and the outcomes are assessed in a consistent way. So there's no bias in terms of the way that the groups are examined and followed and the data collected. Now, we randomize participants to the groups, and there's a very good reason for this. And the first thing is we want to achieve baseline comparability between the groups on factors that could affect the outcome. And the thing is that, and the nice thing about the randomization process is that the factors that might affect the outcome may be known or unknown. And by randomizing, particularly if we have a large enough sample size, the groups will be essentially identical, except for the intervention. If you only have a small group, then they may not be so identical, as we'll talk about. Another approach may be used then. But generally, if you have a large group which you randomize, then the groups will be pretty well equal in terms of all these other factors that could affect the outcome, whether we know about them or we don't know about them. And you can say that everybody, when they are entered into the trial, have the same chance of receiving any of the possible interventions. So this process acts to prevent any conscious or unconscious selection bias. We'll talk about selection bias in some detail in the future, but it could be that physicians have some preference for one or other treatment, particularly in relation to a particular patient, and so by randomizing properly, you avoid that bias. Now, once more I'm going to bring up this word confounding, which we haven't fully explained, but this may give you some sense of how confounding operates. So the thing is that in normal clinical practice, doctors are likely to assess their patients' characteristics, such as particularly the severity of the disease, and they are likely to have a treatment preference, one or the other, based on their clinical experience. Now, if we carried out an observational study of people and just examined patients who were, given one group given treatment A and the other one given treatment B, according to the physician's preference, and we then followed them to see the outcome, whether, say, they were cured, and we might see that one group seemed to have a higher cure rate than the other, but that could have been entirely related to the physician's preference for allocating treatments. So this is called confounding by indication. So it will lead to, say, treatment A appearing to be better than treatment B, but it could have been simply that treatment B was given to more severe patients, who are patients with more severe disease, and so it could be that the fact that treatment A appeared to be better was simply because they were given to patients with milder forms of the disease. So they had a higher cure rate, but it made it look like treatment A was more effective than treatment B. So this is confounding by indication, where the confounding factor is the disease severity or some other patient factor that determined the allocation. And this is a problem in observational studies when you're comparing treatment A and treatment B. It may simply be these other factors. So the great thing about experimental studies is that randomization takes that decision out of the hands of the physician. Basically, the physician allocates treatment A or treatment B according to the randomization process, and it eliminates this problem of confounding by indication. So in the next talk in this series, I'm going to talk about issues such as masking or blinding of participants and investigators, and also talk about the various randomization methods that we can use.